Good afternoon, everyone, and welcome to the monthly FCI seminar series. We are just at the top of the hour, so we will give everyone just a couple minutes to sign in, and then I will introduce Shruti Choudhary. Shruti, I'm sorry, I don't want to butcher your last name. How do I pronounce your last name? Choudhary. So we will be introducing Shruti Choudhary in just one moment. Thank you all for being here. All right, so let's get started. I'm really excited for this presentation and for Shruti to present today on the risk assessment of aerosol emissions during fire training among firefighters. So Shruti is our doctoral student in Chemical, Environmental, and Materials Engineering Department here at the University of Miami. That's under Dr. Biswas's lab. Her current research focuses on airborne transmission of infectious diseases, understanding the interaction of light with matter, development of particle sensors, and air pollution. So without further ado, I will turn it over to Shruti.
Thank you, Tara, for the great introduction. Hello, everyone. Thank you for joining the talk. All right, I will just start with the outline. So the outline of the presentation is as follows. First, we'll talk about the aerosols and its importance, how firefighters are at the risk from bad aerosols, the health impact of aerosol emissions, the past studies and research gap, and the objective of current study, the experiment that we performed, some preliminary results followed by conclusion and the upcoming work. All right, so let's first talk about aerosols. So aerosols are particles suspended in air. They are present in all shapes, sizes, and chemical composition. Just to have a great visualization of how aerosols are, you can think of a hair strand. So the hair strand and your fine beach sand, just look at the picture here. They are of similar sizes, but in air quality, we in general classify particles based on PM concentration. So we have PM10, PM2.5. Now we also have PM1. So PM10 corresponds to the mass concentration of particles having size less than 10 micrometer. So essentially, you can see these tiny blue spheres, which are around the circumference of your hair strand, whereas PM2.5 particles are the particles having size less than 2.5 micrometer. So that will be something more like if you zoom in and then you will see that in one PM10, you will see more of PM2.5. So with the advent of COVID-19 pandemic, you all got to know about the term aerosols, and you thought that they are all the bad ones. But that is not the complete story. The aerosols can be good and bad. Talking from the good perspective, good aerosols are now being used specifically for drug delivery, specifically for cancer treatment, and now they have found application in medicine for the pharmaceutical industry. They have also been used for the advanced material synthesis for energy conversion, agriculture, and industry operations. So that was about the good aerosols. The bad aerosols, the one that would be negatively impacting our health. So that comes certain examples like wildfire and biomass burning smoke. So firefighters are in general exposed to this certain smoke. Then we have some of the problems specifically related to South Florida, that is Saharan dust. Just look at the visibility in these two pictures, which were taken on June 14th and June 20th. So that's like a visual difference. And then also we have the bad aerosols as the infectious aerosols in public, healthcare, and indoor environments. So how firefighters are exactly at the risk from the bad aerosols, because good aerosols are the one which are good for the humanity. So let's consider an example. This is our firefighter, and just this uh, picture is about the respiratory system of a firefighter. Now we know that firefighters are exposed to a large concentration of aerosols. So when they inhale those particles, some will deposit in your head airway region. And this applies to all human beings. So some particles will deposit at the head airway region. Some particles will deposit in your tracheobronchial region as the green region. And some particles will go all the way into your alveolar region as to deep inside your lungs. So this is like a final story of how your particles are depositing inside your respiratory system. But you can, in that case, if I look at how your particles would be interacting with your alveolar region, just to zoom in, you have two different types of picture here. So healthy alveoli, where your particles are not present and it's very easily exchanging the gases with the bloodstream. But in the case of the carcinogenic particles, your alveoli would no more be healthy and it would be damaged. There are other particles as well that would stick onto the skin of the firefighter and also on the firefighter gears. The particles that would stick on the skin, they might diffuse into the skin and then to the bloodstream, which might cause secondary problems. And then the particles that are sticking onto the firefighter gears, if you doff them off, they would again aerosolize and resuspend in the air, which again we might inhale, and then they can get into the respiratory system. So in general, the health impact of aerosol emissions, there has been a link where many studies has shown that your aerosols could have several health issues. Some of them are aggravated asthma, decreased lung function, irregular heartbeats, premature deaths, non-fatal heart attacks. And a recent study, which was about that wildfire smoke impacts respiratory health more than fine particles from other sources. That was an observational evidence from Southern California. So according to the World Health Organization, the worldwide ambient air pollution accounts for 43% of the deaths and diseases from chronic obstructive pulmonary disease, 27% of the deaths from ischemic heart disease, 24% of the deaths from stroke, 17% of the deaths from acute lower respiratory infections, which is of interest to us, and then 29% of the deaths from lung cancer, another interest for us. So in the past, there has been a few studies which had a look at what is the aerosol concentration that firefighters are being exposed to. And those findings suggest that firefighters can be exposed to large concentration of aerosols during live fire training, but also in the firehouses where rest areas contain kitchen, and that the exposure can exceed the EPA national ambient air quality standards. The wildfire firefighters performing direct suppression had statistically higher exposures than those performing staging and other tasks. There has been studies where you could mount your air quality sensors onto a robot to determine the position of the aerosol emissions. So you would find some applications where people are deploying the robots in order to look at the aerosol exposure and the emissions, which brings us to the gaps in the studies. The first gap is that the transport and the fate of aerosol emissions are not well characterized, so that we can eventually get to know how can we reduce the exposure to the firefighters. There has been no study that examined the role of particle resuspension for the exposure to firefighters, which is of another interest to our study. And then there's a missing information about the link between particle inhalation and certain cancers in firefighters. So the overall objective of our study is to understand the transport and fate of carcinogenic aerosols from fire during fire training scenario using a network of low-cost particulate matter sensors and assess the respiratory health risk factors. All right, let's start with the experiment, the pilot study that we did. The figure on the right belongs to the Coral Springs Parkland Fire Department, where we performed a few of the measurements. So the instruments that we used for this study was a low-cost particulate matter sensor and a Grimm aerosol spectrometer. So the low-cost particulate matter sensor was hooked onto the firefighter gear, as you can see on the picture on the top left. The advantages of such a sensor were that they are very easy to use. They send us the real-time data, like every 15 seconds, we get to know what is the aerosol concentration, and they send that data directly to the cloud via Wi-Fi connection. And they are low cost. And as you can see, they are compact size and less bulky. So it's not at all problematic to the firefighters in general. And then we also used a Grimm aerosol spectrometer, which was acting as a reference instrument for the study. And that provided us a detailed size distribution of the aerosols, more in the size range of 200 nanometers to 35 micrometers. Size distribution is something more like what is like number concentration of different size of the aerosols in the environment. There are more sophisticated aerosol instruments available. However, they are not easy to use in a setting like firefighter training. However, we could do much more detailed study in the case of a lab environment for such a scenario. So this is the picture of how the firefighting environment looked like. There were 2 trainings, but one training happened at one time. So like in the case of first training, these blue dots resemble the position of the low-cost sensors that we were using. The oranges region is your hot zone, the light orange region is your warm zone, and then this entire blue region is the cold zone. The wind were north to south, as shown in this diagram. So eventually the sensor, which was in the cold zone, wasn't being directly exposed to the smoke that was coming from these 2 training sites. Okay, so let's talk about the results that we obtained. So here, the figure on the left shows about the red curve, which is all about the hot zone. That is when the sensors were present in the hot zone. The green curve represents the sensor data from the warm zone. And then the blue curve represents the sensor data in the cold zone. Please look at the y-axis, which is different for different zones. Again, the yellow region represents training 1, and the gray region represents training 2, and the white region is where there was no training. So let's talk about the hot zone first. In the case of hot zone, during both the trainings, there was a large concentration of aerosols that the firefighters were being exposed to. However, they all were wearing SCBA masks, so it's not necessarily that they were inhaling those large concentration of aerosols. What the picture shows here is that very large concentration of aerosols were being emitted. Now, if I talk about warm zone, that is where there was no active fire, you could still see a large concentration of aerosols. These aerosols came out when the door from the cabins were being opened up, so the smoke directly came onto the sensor. And then you have a few in the cold zone. For the cold zone, the access is very less. Like here, if you see on the left, it's 2000, whereas it's 5 over here. So you'll see that very less concentration of aerosols. However, there were few cases where the particles from like hot zone transferred to the cold zone, and we saw a few peaks. And then when there was no training, there were some incidences where when the firefighters were doffing off, we saw an again increase in the PM concentration. So as to summarize, particles from hot zone transfer to cold zone at the firefighter training site, and that largely depends on the wind direction. And secondary particles emissions were present during the gear doffing procedure, and that exposure could be higher for firefighters, but that depends on which zone they were working on. So that was about how much concentration were they being exposed to. From the aerosol science, we can actually determine how much of the aerosols would deposit in the respiratory model. So first, in order to understand where particles will deposit in your respiratory system, we need to understand what is the size distribution of the aerosols. So you have two curves over here, the red curve, and the axis for the red curve is on the left, and the blue curve, the axis is on the right. The red curve represents the firefighting activity, like the active fires, and the blue curve represents before firefighting activity. So just look at the difference. There is the red curve goes as high as 50,000 particles per centimeter cube, whereas in your blue curve, the maximum goes 160 particles per centimeter cube. So there's a huge difference between the particles emissions. Again, the second point to note is that your particles were very high in the size range, 200 nanometers to 600 nanometers. So if I look at that region over here and compare that to the respiratory deposition model, I can see that the particles can deposit, like they are very highly likely to deposit in the alveolar region, some in the tracheobronchial region, and some in the head airway region. I also want to point out that we were doing measurements which were very precise in giving us the sizes from 200 nanometers and above, but we haven't looked at the particles which were having sizes less than 200 nanometers. And for that, we need to perform the lab studies in order to understand if there are particle emissions much less than that. Because if the particles are having emissions in between 10 to 100 nanometers, there is a huge deposition of the particles in the alveolar region and in the tracheobronchial region. So they will have the direct impact onto the cells of the respiratory system. So as to conclude, the firefighters are most likely to inhale toxic aerosols without SCBA. Exposure to toxic aerosols from secondary emissions and aerosol resuspension is most likely during geared doffing procedure. Particles with size less than 700 nanometers are emitted during fire activities, which can deposit in the various regions of the respiratory systems, as we saw in the previous slides. And firefighters and staff should wear SCBA in both hot and warm zone, as we saw for the increased concentration of aerosols in both the zones. As a part of our upcoming work, we want to quantize the secondary emissions from firefighter gears before and after the decontamination with water, because that will help us in understanding how much of the particles are actually being removed during the decon procedure. And then finally, we want to study the cytotoxicity study of effects on the skin and the respiratory system cell lines based on different aerosol sizes, because different aerosols would interact with your cell lines in different ways. So we want to understand which size of the aerosols are much more harmful for the firefighters. These are some of the references which you can look over, and thank you if you have any questions.
Thank you so much, Shruti. Super interesting stuff. I'm going to open it up for questions. If anyone has any questions in the chat, please feel free. Um, can you go back to the reference slide again?
Oh, sure.
Someone was asking to see the references.
Okay, just one moment.
And just so everyone knows, we will post these slides as well as recording on our website and our YouTube channel. So in case you don't get to read all of these or get to save all of them, it will be up there. Any other questions we have for Shruti? So I have a question, Shruti. So I know Dr. Caban went with you. I went the next time when you were collecting the air samples out in the field with the training exercise. Can you talk about some of, I guess, just the experience of collecting the aerosols and what were some challenges when collecting it from a live training burn?
Okay, so there were some before sampling challenges and some after sampling challenges, as well as during the sampling challenges. So the before sampling challenge was more about we are going to take these sensors into the fire region, and we are not sure if these sensors would work well in very high temperature environment, because we knew that some of the sensors we would be like cooking them with the firefighter gears, and what if the temperature goes very high and it might impact the firefighter safety. So we quite had a lot of discussion about it, and then we were like, okay, maybe it would not be that of a problem. During the sampling, it was more about what is the best location to place the sensors, understanding how the firefighters work and not intervene into their work was the challenge, because we don't want to like harm or do anything to them. And then some were like, we also wanted to, like, I was more interested in sampling like particles in the size range 10 nanometers to 1 micrometer. So for that, we need much more sophisticated instruments. However, that exercise, it's very hectic, busy, and I was like, I wish I could take the sophisticated instruments, but I could not take that. That still remains a challenge, that how could we take such sophisticated instruments and get the complete data for that. But I do appreciate the work that the firefighters do. That day I was like completely stuck. I was like, oh God, this is just another level.
Yeah. And I think that's so important to really understand their risk and everything that they go through when we're trying to implement something, either for exposure assessment or for protection. You know, that's not cookie cutter like in a lab. That it's always great when our scientists are able to go out in the field and see everything that firefighters go through. So I'm glad you're able to experience that and see some of the challenges that they go through.
Yeah, and then now, like once I performed the first set of experiments, I was like, if we get some more volunteers to work with us, specifically, we're interested in just giving them the sensor, and then just like if they could just stuck their sensors with them, and then we want to see how much is the complete exposure while they are at the firefighting station, because it will be different in different regions. So that is one another challenge, as how can we like do that and then still get the complete exposure to the firefighters, and it would be different for different set of people, but yeah.
Great. Shruti, we have a couple questions. So are there any specific classes of aerosols that are most concerning?
So the class of aerosols, so it now it would depend, like in case of the fire training, in general, it's more the carbonaceous particles. they would obviously, if inhaled, would be dangerous. But if you go to some real firefighting arena, there would be many more particles, and like PFAS, all those things would be in the particle itself or in the gaseous form. There would be VOCs as well, which might be a problem to the firefighters. Like sometimes if you're burning, we never know what toxic elements are being coming out. So there are still needs to be still a lot of studies need to be performed in order to understand much like what other particles could be of toxic to the firefighters.
Great. And someone had a comment, Cindy, great conclusion list to support SCBA use in the various zones. So I think that is something that's really important that came out of this work. Is how important and how well SCBA's work to protect our firefighters from these aerosols.
When I was standing there, I was like I know everybody uses SCBA in the hot zone, but not everybody might use in the warm zone. But looking at that entire situation and what the data shows, everybody should wear the SCBA in the warm zone too. And in some situations, cold zones too. It depends on how your wind direction is.
Absolutely. The next question we have from David is, would you describe the training exercise that firefighters experienced during the sampling? Do you remember what they were doing? So it was a live fire burn. And I believe there was 2 crews that were going through, around 15 minutes per crew that went into the fire. So it was a structural live training burn at a training center.
Yeah, absolutely. Yes.
So the next was, is it accurate to say that the data gathered in the controlled environment burn used for the study would be significantly higher in our real world scenarios? Example of a single family dwelling fire.
Okay, it depends. So if you go to a wildfire, the concentrations would be much higher. But if you are going to just like maybe a single family dwelling fire, it might either be similar or less, but it would all depend on what kind of fire is exactly there. So but if you're using SCBA mask near the fire's activity, like in the hot zone, the exposure would be less, I assume.
What was being burned in training to produce smoke particulates? You know, do you remember what was being burned? We can find out exactly. I don't know if anyone from Coral Springs is on that was there at the training, but we can find out exactly what was being burned. I don't know. I don't have recollection of the exact materials. Shruti, unless you did, but we can definitely pose that question.
Sorry,
find out.
I do not remember that, but I would
and follow up.
Yeah.
So great questions that have come through so far. Does anyone have any other questions? Thank you, Brian. So possibly untreated hay, but no furnishings are allowed. So that's, you know, our guess right now for the training environment, but we can definitely follow up and confirm what was there. All right, so that is all the questions we have so far. So I just want to thank everyone for being here. Thank you, Shruti and the Viswas Lab for putting on these great projects and presenting today. For everyone, next month, same time, we will be doing our next monthly seminar series that will be hosted by Umar Bakali from the Department of Biochemistry, who will be presenting on wildland exposure in our wildland firefighters in Florida. So looking forward to another great presentation. Thank you all for being here. We will be sharing this recorded seminar series as well as the slides on our website. Please also remember, on our website, if you visit, you can see the registration and the save the date for our 2023 International Firefighter Cancer Symposium that will be taking place here in Miami, Florida. Local 1403 has been gracious to host us at their Union Hall and Training Center in Miami-Dade for Miami-Dade Fire Rescue. That will be on February 23rd through 25th. 23rd to the 24th will be the actual symposium, and the 25th will be the Dolphins Challenge Cancer event. So if you're interested in participating and being part of our team, all of that information is also on our website. And anything else that comes up, please feel free to reach out to any of us from the FCI team that we can answer related to any of your questions for your department or any research studies. Thank you all for being here.
Thank you so much.
Mhm.